Welcome back, and thank you for staying for the final session today. I'm Lorraine McElhinney. I'm a co-director of UK-ICN. I'm from APHA.
My name is Jonathan Kotwa. I am from Sunnybrook Research Institute in Toronto as a postdoctoral research associate, and I am the co-chair today.
Thank you. So reminder to the speakers, the first three speakers we have, you'll get a warning at 12 minutes and then 3 minutes for questions. And so first off, we've got Dr. Catarina Nunes from the Francis Crick Institute, and she's going to be talking about RNA packaging, the key to understanding viral assembly. So thank you, Catarina.
So, hi everyone, my name is Catarina and I'm a postdoc at the RNA Virus Replication Laboratory at the Francis Crick Institute. And today I'll be talking to you about RNA packaging and some of the ways that we are using in the lab to understand this key aspect of viral assembly. So I'm not going to go into much detail about the intracellular life cycle of the coronavirus, but I'm just going to highlight what I think is a very interesting phenomenon that occurs in which in the replication of coronavirus, there is the production of the full-length genomic RNA, as well as the subgenomic transcripts, and at some point they all coexist in the cytoplasm. However, when the virus then needs to package, it needs to selectively package the full-length genomic RNA and not the subgenomic transcripts. And exactly how this occurs and how this is regulated is the main topic of today's talk. And for that, we focus on the later stages of viral replication, from virion assembly to viral egress. So what we know about packaging comes mainly from MHV, and using P32 labeled virus, the authors could detect that, and when they did like fractionation or sucrose gradient, they saw that they could detect specific packaging of some defective interfering RNA particles together with the full-length genomic RNA. And by analyzing the sequence of this defective interfering RNA particles in comparison to others that were not successfully packaged, they managed to map the packaging signal to a specific region in the NSP15 gene. Then further mutational analysis and computational modeling led to the consensus that there is this 95 nucleotide bulge stem loop region in the NSP15 that acts as the packaging signal, and that is what is recognized at this later stages of viral infection. So then to understand exactly what was the role of this and if they could indeed validate it, the authors generated or induced 20 codon silent mutations in this bulge stem loop region and created what they called a SILPS mutant, for silent packaging signal mutant, in which they maintained the protein, but they completely disrupted the structure of this bulge stem loop. And when they did the phenotypic characterization of this mutant, they then saw that the SILPS mutant has plaques that are indistinguishable from those of the wild type. They grow to the same extent, but they have problems in packaging, as you can see by the incorporation of subgenomic RNAs together with the full-length genomic RNAs when looking at the purified virions. So we know a lot about this sequence and its structure, but how exactly it is recognized is still a matter of debate, and there is some ideas around. They're not mutually exclusive, and some authors suggest that it is the N protein that binds to this packaging signal and then interacts with M. Some others say that M is the main interacting protein with the packaging signal, and yet some others say that N and M form a complex, and then this is the complex that binds to the packaging signal and then leads to the recognition of this signal. So this then leads us to the main open questions of our project, in which we're trying to understand how this packaging signal is recognized, if it is functionally conserved in other coronaviruses, and if the NSP15 packaging signal that was identified in MHV is both necessary and sufficient to regulate efficient packaging. And today I'll be talking mainly about the question number 2, in which we're trying to see if this is conserved in other coronaviruses, specifically in the human beta coronavirus OC43, which is which is related to MHV, but it infects humans and causes mild respiratory disease. So when Harriet Mears was in David Bower's lab, she did some RNA shape. And what you can see here is that she detected that for OC43, and in comparison to MHV here on your right, she could detect this structure that is this bulge stem loop around the NSP15 region. And it also has this repeated sequences of GU-RAU motifs, similarly to what she could see for MHV. So there was this structure and some sequence similarity between OC43 and MHV already. However, we wanted also to understand if we could see if other viral proteins such as N and M could interact specifically with this structure in OC43. So for that, we have been using cross-linking immunoprecipitation in sequencing, or CLIP-seq. And the way that this works is that we have our purified variants and we expose them to UV light for cross-linking. We then do immunoprecipitation using antibodies for our proteins of interest, the ones that we want to see interacting with our RNA molecules. So in our case, for this project, we use N or M. We then do an adapter ligation and proteinase K digestion. And so what happens then is that when we are doing our library prep, our RT will stop at the cross-linking site. So we'll end up with some fragments that are always truncated at the protein binding site. And so this then can be interpreted as a peak for a region of the genome with which the proteins are interacting preferentially. What you see here is the nucleocapsid binding across the genome through that data that we obtained through CLIP-seq. And you see on the y-axis is the cross-linking signal per million, and this is the genomic position. And you see the data both for OC43 and MHV. And you see that N binds across the genome, but if we look specifically at this region at 20,000 nucleotides, which corresponds to the area where NSP15 is, you see that both of them show a peak of association of N to that area of the genome. And indeed, if we zoom in with a 20 nucleotide smoothing window, you see that there is indeed a peak of the association of N with this region in both OC43 and MHV. And this corresponds to the region which Harriet had identified as this bulge stem loop in her shape data. So now we had this structure similarity. You had the repeated motifs. We had other viral proteins that were interacting with it. The question is, can we disrupt it? So by using the reverse genetic system that was established in the lab by Kieran Gilbride, we induced some codon shuffling mutations in the NSP15 region. And again, the idea is that we maintain the protein itself, but we disrupt completely the structure of this bulge stem loop in this area in OC43. And then I did the characterization of this mutant by doing growth curves, both by plaque assays and qPCR in lysates and supernatants. And what you can see here, so in the y-axis, we have the PFU per mil over time in a low MOI infection, and you see three lines. You see the wild-type virus, you see our CLPS mutant, and you see an MNG that we are using as a control for our reverse genetic system.
Sorry.
And you see that there is no difference between any of the three viruses. And the same thing is true when we look at the expression of NSP12 and M, both in supernatant and lysates. And you see that there is some variation at initial time points of infection, but these kind of dissipate later on. So there doesn't seem to be an impairment in growth for the CLPS mutant. However, when we looked at the plaque phenotype, you can appreciate here that CLPS actually has smaller and less well-defined plaques than the wild type. And you can see that quantified here on the right with the plaque area in the three different viruses. And in this case, the different letters mean statistical significant differences. So although there is no difference in the replication in viral growth per se, it does seem that the CLPS might have compromised cell-to-cell transmission due to packaging. To understand if you could see any disadvantage for this mutant in any other conditions, I then did a competition assay in which I co-infected cells both with wild type and CLPS, and I identified the different viral titers throughout time by using probes with locked nucleotides at specific sites where mutations were. Again, if we now compare the viral titers both in the supernatant and in the lysate, it does not seem to be the case that SIL-PS is outcompeted by OC43. So in summary, I showed you that MHV packaging signal, identity, and function seems to be conserved in OC43. Our ClipSec data shows that M and N binds across the genome and specifically around the PS. So today I only showed you data on M for the sake of time. Same thing with the data that we have that reveals that there are other new putative packaging signal in OC43. So we have data for this, and the reason why it's here is because it's important for our future perspectives. I showed you that OC43 does not seem to have a growth defect even under competition pressure, although it has signs of attenuation, particularly with the smaller plaque phenotype. So in the future, we would like to explore these alternative packaging signal that the ClipSec identified in OC43, try to understand if they can contribute for packaging in coronaviruses, and even if they can offer some sort of compensatory or redundant effect in our SIL-PS mutant phenotype. And with that, I would like to thank the entire Bauer Lab, and in particular David Bauer, Harriet Mears, that is really the engine behind the project, and she's now in Bristol running her own lab. Anab Chakrabarti for his help with the ClipSec and all the ClipSec analysis, Kiran for the reverse genetic system, and Zi Yang for help with the shape map data. And thank you all for listening.
Thank you so much, Catarina. Any questions?
Thanks very much. Very interesting talk. Presumably your growth curves are done at quite a high MOI, are they, of the virus?
So we've done it both at MOI of 3 and 0.01.
Okay, because I was going to ask, because your plaque type, if you start at a very low MOI, would you then find the spread much slower with your small plaque variant?
With the small plaque, can you repeat that, please?
If you start at a very low MOI,
yeah,
would you find that your spread is slower
yes.
than your large plaque variant, and might that be a disadvantage?
It's not really. So the data that I showed here is the 0.01 MOI, which is the lowest that we've tested, and they seem to be, as you see in the growth curve, they seem to be quite, they grow quite indistinguishable from each other at that MOI at least. I haven't tested lower than that.
Okay.
Anyone else?
Hi, thanks. I just wondered, in the particle compared to intracellular RNA, could you get differences in what you think is a packaging signal?
Compared to?
Well, when you've looked at the virus particles, right, for the ClipSec?
Yes.
So if you compared that to when the RNA is getting packaged in the cell.
Yeah, so that is, so as I mentioned, we're doing this with purified virions. Obviously, one of the things that we've been discussing is like what happens if we then try to look at a stage where we still have packaging occurring. So you would have to do like self-fractionation for that. It's not something that, so we are aware of it, but it requires a lot of material to do the ClipSec. And by the time when we were like optimizing it, the easiest and fastest way to go was just to go straight to the purified virions, also to validate the signals that we're having. But yes, we are aware that that is something that we should keep in mind.
Thank you, Paul.
What do you see inside the mutant virus particles if you just isolate the RNA and run it on a gel? You start seeing the subgenomics being included.
We haven't done that. We thought about doing northern blots, which is the same way that it was done for the MHV ones, but we still haven't really optimized how to. It's a lot tricky to put like the entire genomic RNA into the northern blot. We haven't managed to find a way to do it in a clean way. But yeah, that is a very good point.
Thanks, everyone. So thanks, Catarina. Moving on.